Good afternoon to everybody, and welcome to this Leading Lights lecture, or inaugural lecture if you prefer it more traditionally, for Professor Mark Pallens, who it's my pleasure to introduce today. I'm Peter Winstanley, for those of you who don't know me, and I'm the Dean of Medicine here at the University of Warwick. I extend the warmest of welcomes to a very large family contingent today. To Helen, who is Mark's wife, and to their children, Emma, Charlie, Tasha, and Tom. Mark's parents, Mike and Deirdre, are also with us today. We're particularly delighted to welcome quite a bevy of former teachers and colleagues, particularly Jim Mangum, Mark's old biology teacher, and I've already told him that this is Warwick, so he doesn't get a cut of any of the grants that Mark is expected to win over the course of the next few years, and his wife Beverly. So Tabak Charlie, who inspired Mark as his professor at Bart's sub-bibliola years ago, I think. Brendan Wren, Nick Lohman, Charles Penn, Steve Busby, and John Heath are all with us today as well. Welcome to you all. So Mark Pallens was an undergraduate at Cambridge and then went on to obtain his PhD from Imperial College. He qualified also in medicine at Barts and the London initially, and then went on to become a fellow of the Royal College of Pathologists, which includes medical microbiology, even though it doesn't necessarily mention it. Mark joined us at Warwick almost exactly a year ago from our neighbours at the University of Birmingham. His research interests focus particularly on bacterial pathogens and communities of bacteria, and he's interested particularly in the translation of research findings for the benefit of patients and the public. He's at the cutting edge of efforts to use and exploit high throughput genomic sequencing in medical microbiology, and I've no doubt he'll tell you more about that today. Earlier this year, we were able to announce that Mark and his team had won a major MRC competition for a centre in microbiological bioinformatics, a total of £8.5 million, which is shared also with the University of Birmingham and Cardiff and Swansea. And this initiative is going to build new ways of linking across complex biological data and health records to solve key medical challenges. Mark's got over 140 publications listed in PubMed in major journals, including JAMA, The Lancet, Nature, and many more. He quite literally wrote the book on evolution, having authored the rough guide to evolution, which some of you will have seen. So one or two of you might be unfamiliar with universities, and so I should sort of describe what universities are all about. Those of us who are aficionados know this already. A university is a group of squabbling and competing academics who are unified by their common grievance over a lack of car parking, and I'm very surprised indeed if Mark says no comments in about car parking today. So I'm delighted to invite Professor Mark Pallens to present his inaugural lecture entitled "Nothing in Microbiology Makes Sense Except in the Light of Evolution." Thank you.
Okay, so that's the title. Before we get stuck into the body of the talk, I'd just like to add a few online extras. So live tweeting is welcome, and there's the hashtag for it. The slides are already available on SlideShare. There's an online guide So some of you probably will have this kind of ADHD where you can't just listen to me, you'll be on your phones and tweeting, and there's one just doing it now. You can follow all of the sources of what I've said and refer back to information using that, or you can do it at home if you like. And this is being live streamed to the YouTube channel of the medical school. So it's a bit like on those quiz shows where they say hello to all the listeners on Dave, hello to anyone who's out there watching us on YouTube at the moment. I'm releasing the words coming out of my mouth and the slides under Creative Commons license. The university is retaining the copyrights of the video. And just in case anyone doesn't know, we're going for drinks afterwards at the Varsity Pub. I'd also like to put this lecture in memoriam of 3 individuals who've had an influence over my life, I suppose, and over the academic disciplines in which I dwell. I'm not going to say more about them, but they've all died in the last few years, and it will become clear what their influences are later on. And I have to get the acknowledgements out of the way first. So I acknowledge all the people I'm currently working with, all of my research group. I'm not going to name them because we don't have time, but again, if you look online, you'll find all that information. And similarly, I'd like to acknowledge all the people who have influenced the trajectory of my life, my academic life, my intellectual development over the years. And again, I'm not going to go through and mention all of them. Peter has already mentioned some of the people who've actually come here today. I'm very grateful for that. Another thing I have to say, let's get this out of the way as well, is that I'm not obsessed with Darwin. Okay? Despite quoting him at my wedding, calling 2 of my children Charles and Emma, being the first name terms with 2 of his descendants, writing scholarly works and Susan. I know other people who are. John Van Wy in Singapore, he's definitely obsessed with Darwin. Alison Byrne, who works for the Darwin Correspondence Project, she's obsessed with Darwin. Randall Kane, Darwin's great-great-grandson, he's obsessed. I just have a passing interest, and I'm over that now. But it's a bit like if Paul McCartney were to do a concert and he just did the front chorus. The audience would be disappointed, they're waiting for Hey Jude. So I'm going to have to put in a bit of Darwin, I can't help it. So there will be some of that as we go through. So what am I going to cover? I'm going to cover the history of life, history of the universe really, history of science, history of me and my science, century by century, decade by decade since the beginning. Now you might say, well, that's rather a hard undertaking. You've got less than an hour to do this. It's simply not going to be possible. Well, we have to do what the mathematicians call simplification. We're going to make a simplifying assumption. Simplifying assumption is that the world, as we know, was actually created on November 24th, 1859, at the time that Darwin published The Origin of Species. And it was endowed by its creator with the semblance of a long history that goes back billions of years. That was all just a ruse, and actually came into existence on that particular date, in 1859. So that makes it easier. We've got a century from 1859 to 1959 to cover. Then we can go through the various decades since my birth in 1960, and it should be therefore fairly straightforward. So 1859, the first thing we have to talk about is obviously Darwin and Darwin's influence on modern thought. And just to recap here, I'm drawing on an essay by Ernst Mayr, who is one of the kind of godfathers of evolution, lived to be over 100 years old. And he basically distilled out of all of Darwin's work a number of ideas. He said that Darwin basically gave us the notion of branching evolution with the common descent of all living things, told us that the mechanism of evolution was largely, not exclusively, natural selection. That evolution was gradual with no major discontinuities. And he pointed out that evolutionary biology is, unlike, say, chemistry, is a historical science. In evolutionary biology, you're looking to reconstruct what's happened in the past and come up with narratives to explain how things have come to be the way they are. The other interesting point that Mayr pulled out is that basically evolution feeds on variation and randomness. And it banishes this kind of typological thinking where you can capture everything in one particular instance, and also banishes determinism. So for tidy minds, this is a bit of a problem. You like everything to be the same. Physicists say that one electron represents all electrons in the whole universe. but we as biologists recognise that one organism is going to be different from the next organism and the next organism all the way through the population, pretty much. Now, while we're thinking about Darwin, I'm just going to bring in one other thing that I will come back to, which is that it's often difficult to actually do your thinking purely in your head. And sometimes you have to have tools outside of yourself to use to actually do thinking, research, and so forth. And Darwin actually did this in a big way with his notebooks. And he's kept these very complex and detailed notebooks. And we have these, and they're all available online. And it's one of the great pleasures of writing the Rough Guide to Evolution is I actually force myself to go and read them all, and there's some wonderful things in there. Here's a sketch there that he drew of an evolutionary tree, one of the first trees ever drawn to describe evolution. And you can see the dotted lines are the extinct things, and then the solid lines are his idea of current species. And his thoughts range very widely over all sorts of things. A man in Shrewsbury who used to call people bastards when he was drunk, and someone who tried to have sex with a bird, and all sorts of weird things. In fact, I have a whole talk called The Rough Guide to Darwin, where we go into Darwin's dirty, as we might call it. But I'm just going to share with you this one quote here, which I think is particularly poignant, where he says, "If we choose to let conjecture run wild, then animals are fellow brethren in pain, disease, and death, suffering, famine, our slaves in the most laborious works, our companions in our amusement." And then he kind of makes this point that we may partake from our origin in one common ancestor, we may all be netted together. And in a sense, that prefaces all of the animal rights movement and the interest in ecology and sharing the world with these animals, and also vegetarianism, if you like. So what else went on? Well, around the same time, Mendel was doing his experiments on peas, and the discipline of genetics kind of booted itself up rather belatedly. So Mendel's experiments they were forgotten about and ignored for several decades, and then around the beginning of the 20th century they rediscovered various other insights that came into genetics, the difference between serma and germline and chromosomes and mutations. And then in the 1940s, evolution and genetics were fused together to what's called the modern synthesis, with various architects there, including Dobzhansky, who we'll hear more about later. Okay, that's one thread. So we've got evolution and genetics. Another development that was going on around that time, in fact predating, I've kind of gone back in time here, before 1859, but in the 1830s, Babbage was working on the analytical engine, first modern, if you like, computer. Ada Lovelace, Byron's daughter, wrote the first program for it. In fact, Darwin met Babbage at dinner several times, I think. Boole invented Boolean algebra in the 1850s. I put in some stuff about, just a mention of Turing here for John Heath. He's obsessed with Turing, designing Darwin. And then the 1950s, we started to see computers as we know them. But probably the largest conceptual leap forward, the one with the most impact, was the idea of germ theory, the development of germ theory, which followed Darwin's species a few years later, sort of kicking off with Pasteur demolishing the idea of spontaneous generation in the 1870s, Koch linking anthrax to bacteria, Ferdinand Kohn, German bacteriologist, founding bacterial taxonomy. And it's worth noting there, we're going to root everything in Darwin, that Kohn and Darwin actually had quite a lively correspondence, and Darwin was kept up to date with the developments in microbiology through that correspondence. And in the 1880s, TB, cholera, diphtheria all linked to germs, to bacteria, anti-serum. Two papers there which are particularly worth noting, from Koch and Graham. Koch, first of all, pointing out in 1881 that it was a good idea to grow bacteria on a solid medium, and you could get colonies and you could propagate them in pure culture. And Graham pointing out, look, you could stain them in a particular way down the microscope and differentiate them and identify them. And I mention that because actually not much has changed in microbiology since the 1880s. If we fast forward now to the 1940s, Julian Huxley, who was the grandson of Thomas Henry Huxley, Darwin's bulldog. There's Julian Huxley on his grandfather's knee. He was quite strident in saying, well, bacteria, they're not real organisms, they don't have genes in the normal sense. You know, genetics doesn't apply to bacteria. So he put his head above the parapet, and he was wrong, but that's what they said. Then you fast forward another decade or so, and you get to Dobzhansky, one of the architects of this modern synthesis, and in his 1951 edition of his book, Genetics and the Origin of Species, he then points out that yes, there are genes in bacteria, and you can show mutations, and you can. And in fact, the first mutations were described as as antibiotic resistance mutations, and so this brought bacteria into the fold of genetics and part of that modern synthesis, if you like. So another interesting thing happened in the 50s, which was that the structure of DNA was revealed. But again, as we have to keep harking back to Darwin, there's an interesting factoid here for those of you who are interested in such things. It turns out that Darwin wrote his last paper, research paper, it was published just a few days before he died in 1882, in April 1882, and in that paper he thanks a certain William Drawbridge Crick of Northampton, who had supplied Darwin with information on the spread of a bivalve mollusk by being stuck onto the leg of a beetle. Darwin was obsessed with the idea of how do things get dispersed around the world, how do they move, organisms, how do you actually account for their movement. So he was very interested in that and wrote up a little piece thanking Crick. Now it turned out that Crick had a son, who then had who had a son himself. So William Drawbridge Crick was in fact the grandfather of the Crick that wrote the Crick and Watson paper that came out in April 1953 and revealed the structure of DNA. So everything does connect. Now, 1960. Now that was the year I was born. You can see some of these family photographs. Here's my family here having lunch on the new Coronation Street soap opera that they starred in. Here's my mother and one of her friends reading Lady Chatterley's Lover that year. There's my dad just returning from his military service. It's like to do that kind of thing in those days. And anyone know who this person is in the corner? Peter Medawar. So Peter Medawar got his Nobel Prize that year for work in immunology. And as an aside, we want to show that there's an obsession with Darwin. In fact, Peter Medawar basically showed that you could explain all of immunology through evolution. So nothing in immunology makes sense except in the light of evolution because of clonal selection theory, which is a variant of Darwin's selection. 1964, we come to the source of the title for this lecture, which is that Dobzhansky wrote this piece in the American Zoologist. And there are two interesting features here. So on one side here, you can see he's fussing about the fact that molecular biology was taking over. And it's just amazing to me, both here, I think, and certainly in Birmingham, people in schools of life sciences or biosciences are all worrying about how do we keep organismal biology alive, whole organism biology. Everything's becoming molecular biology, or here it's all becoming synthetic biology. And you know, anything to do with animals and plants, real things like that, that are large and you can see them, that's all getting squeezed out. And so it's interesting that this is not new. This is going back 50 years. People are fussing about this. But also he makes this generalization here where nothing makes sense in biology except in the light of evolution, or subspecie evolutionists. That's aping Spinoza there. And so he's just pointing out that basically life is a joke if you don't take evolution into account, its fundamental unity. And he then turned that later, a few years later, into an essay, in fact. So now I'm just going to take a step out of this chronological narrative, and just for those of you who are not biologists, just to point out that a lot of the talk's going to be about sequences. And basically we have two kinds of sequences in living organisms. We have those that are to do with information storage and those that are turning information into action. So we have genes that make DNA, and then we have proteins, and the DNA sequences encode those proteins. And if we take all the genes, all the DNA in a bacterium together, we call that its genome sequence. It's kind of complete genetic blueprint, to use a truism. So I've said all that because in 1965 another breakthrough came when Zuckerkandl and Pauling wrote a paper where they actually pointed out you could use biological sequences to understand evolution and actually draw up what they called a molecular phylogeny. So it was a very eventful paper, in some ways ahead of its time. It's also interesting to note how things have changed again in the last 50 years, because they actually quote Hegel in their introduction. That's the opening paragraph there, and they go on that using Hegel's expression, we may say that there is no other system that is better aufgehoben. I don't think anyone would write a paper like that these days. Mark Uckman has pointed out they misuse the word aufgehoben there, but we can talk about that maybe later in the part. So what they did basically was take this idea of homology. So many of you will be familiar with this, the textbook idea of homology, that you look at the disposition of bones in the limb of a tetrapod, and you can see that there's an exact correspondence. Even though those limbs are doing quite different things, they obviously have similar kinds of structures and morphology there in a kind of deep sense. And in looking at, say, that red bone there, you could argue that, okay, so we've dissected it in a human, we know which nerves go there, which blood vessels go there. We could make predictions as to what nerves and blood vessels would serve it in, say, a bird or a whale, even though it's a different context. So what Zuckerkandl and Pauling did was say, well, actually you could do the same thing with sequences. If you line up the sequences, do what we call an alignment, you can look for things that are the same or similar in two different sequences, and you can look at these patterns of similarity and dissimilarity and draw conclusions about evolution, and more generally draw conclusions about the function of the things that you're looking at. Let's just take another side step and spend a bit more time thinking about this, because it is an important point. So you can make an analogy, in fact Darwin made this analogy, that you can compare the evolution of life with the evolution of language. And now that we understand about sequences, biological sequences, we can make this even more poignantly, because both languages and informational macromolecules like DNA and protein consist of sequences of characters which change over time. They acquire small changes with each generation and slowly but certainly diverge. And you can look at these patterns of similarity due to descent from a common ancestor, i.e. homology, to infer function in the biological sense or meaning in the linguistic sense. So if you look at these 1, 2, 3, 1, 2, 3, and 4, you could say, well, actually the first two sets of numbers there are more similar than the second set of two, and therefore you might want to classify those as more closely related and say the first two are Germanic languages and the second two are Romanic or Latin-derived languages. And in fact, you can do this way back into the distant past. So here's an example of Hittite. And okay, there's supposed to be an animation there, but it's not work. So you're not supposed to see the answer. But this is going to be a bit of audience participation, see if you can recognise any of the words there. But this guy, Bedrick Crosney, back in 1917, came across this Hittite inscription and recognised the word Ninda as an ideogram, a logogram from Sumerian that meant bread, and then just looked at the other words and was surprised to see this water in the next line, and he actually then translated this as now you will eat bread and now you'll drink water. So it's looking for these kinds of patterns of similarity and also looking for overlapping clues. So the fact that there was bread in one word, in one sentence, and then water in the next, and there were two verbs that were in the same thing, that all made sense. Got a life of its own, this, I'm not making it go forward. What's happened here? Anyway, so the point is with language we can go back 3,000 years, but with biological sequences we can go back 3,000 million years, 3 billion years, back to the last eukaryotic common ancestor. Anyhow, in the 1970s, Sanger invented DNA sequencing, so up until then people have been largely sequencing bits of RNA and mostly proteins. But he came along and invented this method of sequencing again, which stood the test of time for 30 years or more. Basically, people still using this kind of approach. And even today, the approaches we use are variations of the same sequencing. Okay, what about me? So, in the 1970s, I went to this place here, Wallington High School for Boys. There's a picture of me. There's a picture of my biology teacher, as he was then, 35 years ago, 36 years ago. A couple of quotes there. One was from my school report, I think from the maths teacher, that said, must learn to control his grasshopper mind. I do struggle to control my grasshopper mind, but I've managed to somehow make use of it. And there's a quote there which I thought originally came from Huxley, but in fact when you look, it doesn't. It's been misattributed to Huxley. It's from an American journalist called Sidney Harris. But basically the primary purpose of a liberal education is to make one's mind a pleasant place in which to spend one's time. And I think that basically sums up my education. I really didn't enjoy the time I spent at school, and I still look back on those times with fond memories and call upon all that. Now interestingly though, I actually got my old school textbook out and had a look at it. And you go into the index and there's no DNA. It doesn't mention DNA in a biology textbook. So that's quite remarkable, I think. It really is full of lots of morphology of insects and plants, bryophytes, all that stuff, but no mention of DNA, no mention of sequences in there at all. But interestingly, I did do the Cambridge entrance exam in the 7th term of the 6th form, the 4th term of the 6th form, and the question that came up in the biology paper was to take a load of peptide sequences and assemble them into a protein. So that kind of, you could say that set the tone for the rest of my working life. After that, I went to Fitzwilliam College in Cambridge. I put a couple of quotes there. Those were actually from Darwin about his time in Cambridge, but they pretty much apply to my time in Cambridge as well. I did have some good times there, but I don't remember much of the course being particularly interesting. It was much more having discussions with fellow students about developments in the field. There's a guy, David Simmons, who's actually unfortunately not here today. He was much cleverer than I was and much more dedicated to his studies, but we used to have interesting conversations. And one time I said to him as we were eating dinner in college, so how much genome do you think there is in this pork pie? But that went too far for him. He said, oh, that's an absurd question. I'm just a stupid person to asking silly questions like this. But of course, now with the horse meat scandal, that's exactly what people are asking. You know, they go and take the DNA out and they say, okay, there's horse DNA, but there's also something else DNA in there. So I also did a final year project on leprosy, which was my first interest, I guess, in infectious diseases and microbiology. And then I quickly bounced around, went to the London Hospital for a while, went to the Royal Free for a while, and ended up at Barts Hospital in London. Now, late 80s, basically there was this, for me, one of those kind of eureka moments when I went to the library. You still had to go to the library in those days, no internet and stuff, and found this this article by Carl Woese on bacterial evolution, a review article, and it basically summarised all that was known about bacterial evolution, and he actually proposed that there was 3 different kinds of life. So the idea of just being bacteria and eukaryotes like we are that have a membrane-bound nucleus, he said, well, actually there's this other group called the archaea, which are just as deeply branching as the other two, and he came up with this kind of trichotomy of bacteria, archaea, and eukarya. So really, this for me was at that time I was already interested in Darwin and evolution and biology, and wanted to see this joining of bacteria and evolution, and it was a great joy to read that paper. But of course, there is nothing new under the sun. If you look at that tree that we draw from Woese's developments, and you go back and look at Darwin's first sketch of an evolutionary tree, you can see they're very similar. And in fact, if we apply this special antidote to the invisible ink that Darwin used in his notebooks, you can see that Darwin did actually get there first. Woese's tree a long time before he did. So as the 1980s turned into the 90s, I actually used that as an opening line in a grant proposal. I was told I'd be a pompous twat by my As the 1980s turned to the 1990s, it was a great time to be working inside the bachelor's department with Brendan Wren, who's sitting there with her. It was a fantastic time in my life. It was a time where we first started to be able to use computers to find sequence homology, and this Apple Mac was the first computer that I actually really used for a long period of time, and it transformed the way we were doing research at the time. So in the group at the time, one of my colleagues, Chris Clayton, had cloned a urease gene, or thought he'd cloned a urease gene from a bacterial host from Helicobacter pylori, but he wasn't certain, and I used a computer, did some searches, and said, yes, definitely, you do have the urease because it's 50% the same as this plant urease. And this convinced me that you could actually make a serious contribution to laboratory-based research by using computers, mucking about with computers. So I kind of redeemed my reputation from that. And then we went on to look at some other genes as well, gene clusters. So some butanol fermentation genes from Clostridium difficile. And again, having this grasshopper mind, I got very excited about that because I recognised that those genes that we got from C. difficile were in fact very, very closely related to genes from another organism called Clostridium acetobutilicum. And that pathway that those genes encoded was used by Chaim Weizmann to actually produce butanol during the First World War and actually helped the British war effort. And that earned it the Balfour Declaration, which laid the seeds for the formation of the State of Israel. So you can see everything connects again, if you've got a grasp of the mind on nice way of thinking. We also found a reverse transcriptase, the group 2 intron at that time. And I started to get more and more interested in this idea of looking for distant homologies and looking for relationships between proteins and their constituent domains that were not quite obvious, that you had to scratch beneath the surface. But when you found them, it became obvious that they had some kind of meaning and could actually allow you to infer commonalities in structure, function, and so forth. And it was a bit like doing a big kind of molecular jigsaw puzzle. You're looking for all these clues that kind of intersect. As we move into the 90s more, though, stress became a big theme in my life. I basically went off and did a PhD, working for Gordon Dugan at Imperial College. And at that time, people were interested in stress responses in bacteria because they realised that actually, when you grow a bacterium on a plate in a lab, it's a very artificial kind of thing to do. E. coli never has it quite so easy as it does in a lab growing on a plate, same as salmonella. And in the real world, they have to fight for a living. And so there was a lot of interest in saying, well, what happens if you starve them? What happens if you heat shock them? And so forth. So I got my PhD. I did spend 3 years full-time at the bench. I went through the period of being the complete lab idiot and not knowing how to run a gel or hold a pipette, but I made good on that. And then shortly after that, I got BBSRC grant to continue this work and actually flourished quite well in this work. Starvation stress for a while. It was also a time of stress because I was on television as part of the Imperial College University Challenge team, and I actually captained the team that won that year in 1995. So that was stressful in a way. Also stressful as that was when we began our family, and those of you who have young children will know that young children bring a certain degree of stress. That stress doesn't actually go away in like 10 years. In fact, it doesn't go away. But I'm very, very pleased to see my 4 children here, and I'm very proud of all the pleasure that they give me as well as the stress. As we got into the middle of the 1990s, we saw the birth of what we might call bacterial pathogenomics or pathogenomics. And I remember one of my colleagues, Duncan Maskell, coming into the lab breathless, having come from a meeting, we all gathered round and listened, and he told us about this guy called Craig Venter, who had sequenced the genome of a bacterium, Haemophilus influenzae. And then to show that he wasn't just a one-trick pony, they'd sequenced another one, Mycoplasma pneumoniae, in double quick time. Mycoplasma genitalium in double quick time. And this was a real revelation. When we started seeing all these genome sequences coming out of bacteria in the late 1990s, we realised that what we'd known about before, discovered biology before genomes, was really like the tip of an iceberg, and there was this very undiscovered biology. So even in an organism like E. coli, that people have been studying for getting on for a century, over half the genes were only discovered when they actually sequenced the genome. We didn't know they existed until we got genome sequencing. And I actually made a good living for myself in this area. Got involved through Brendan in the first Campylobacter jejuni genome sequencing project. And with Brendan wrote a review in Nature on the subject. ASM, the American Society of Microbiology, asked me to edit a book for them. I got involved in a couple of other projects with the Sanger sequencing genome. So it was a very exciting time to be in the subject. But also, around that time, I was also thinking, well, we need better tools to think with. So coming back to what we said about Darwin's notebook, you sometimes need to get out of your head and actually look and use other things out there. So during my PhD, I got familiar with the internet, and I approached the British Medical Journal and said, well, do you want me to write something about the internet that might be useful for healthcare professionals? And they said, yeah, okay. So I wrote a series of articles, which then got turned into a booklet, which then got translated into various languages, and I think you can still buy it on Amazon even to today, although it's well out of date. And one of the ironic things in there was, I said something to the effect that, oh, it's much better to look at email than snail mail because you never get any junk mail through. And we set up these databases as well. So to analyse all these genome sequences, even when there's only a few genome sequences, it was you can't just look at the A, C, and G to make sense of it. So we tried to provide these tools, and Roy Chowdhury, who's the first author of both these papers, is sitting up there at the back, we built these databases, and we built quite a following in the scientific community and helped the scientific community understand these genomes. As we got towards the end of that decade, I got itchy feet. I moved from Berks, then went to Belfast for a short time, and then ended up in Birmingham. And came up with a whole new group of colleagues and new challenges and new interests as I settled in there. So one of the first was coming up against E. coli as a model organism. Steve Busby, who's somewhere in the audience, can't see him for looking, but he's, there he is over there. You know, he basically spent his whole life looking at this one strain of E. coli. And basically the view of people who work in model organism biology is, well, God created this organism, it's not no ecological context, no evolutionary context, it just is. And so I actually got quite cross about this and said, well, you know, it's actually an artifact. And I wrote a piece with John Hodman and Charles Kane where we said these are not really model citizens. These are this E. coli that had been propagated in the lab for decades. It was a deceitful, delinquent, or disgraceful. And we pointed out that, you know, if you want to talk about real biology, you have to talk about fresh isolates that have come out of the real world. At least as well as your model organism. Perhaps one way to make this sound more impactful is, this is say E. coli in a state of nature, fresh-faced and young-looking. Then it gets into the lab and it starts to degenerate a bit. And before long, it's actually achieved the status of becoming an icon. And that's my view of some of the work that's been done on model organisms, and we have to be careful with that. Did do a lot of work over those years on looking at evolution in E. coli, looking at, so we were surprised to find that even in E. coli we could find remnants of systems that no longer work. There was this idea that, well, bacteria were small and perfectly formed, they had these compact genomes and everything in them made sense in terms of current biology. But we showed that wasn't the case. And we also showed there were many ways to get to the same outcome. So a particular kind of E. coli called enterotoxigenic E. coli, it turned out that basically all it required was a toxin gene to come in on a plasmid, and any old E. coli could become toxigenic. Again, these overturned our prior assumptions. I also got interested in a system called type 3 secretion, and we looked at the virulence effectors. So this is basically like a molecular syringe that injected things into eukaryotic cells. And if you were an engineer, you'd design it all nice and compactly so you'd have everything together in one place. But what we discovered wasn't the case at all, that actually the effector genes, the things that do damage, the encoded proteins that do damage, were scattered throughout the chromosome of E. coli, and they were actually present on these mobile genetic elements called bacteriophages, or popping in and out of the genome. So again, this was a surprise where we could see that evolution was a different kind of way of doing things than what we would have designed if we were actually engaged in some kind of synthetic biology. That also sparked an interest in complexity. So closely related to those type 3 secretions I just mentioned is the bacterial flagellum, which is the main organelle of motility in bacteria. And there were some wrong-headed people particularly in the US, who were claiming that the bacterial flagellum was so ornately designed and so irreducibly complex, it must have been designed by a designer. It couldn't have evolved. And so we spent some time debunking that, but also generally looking at evolutionary relationships in this system. I also started running these Darwin Days once a year on Darwin's birthday at the University of Birmingham, and it became a bit of an obsession, I have to admit. Now I'm in rehab, I can say but it did become a little bit over the top. But it was also a great reward, and I got a great deal of pleasure out of this. There's me drinking champagne with Alice Roberts off the telly, who I recruited to a job in Birmingham. There I am with Randall Kane's Darwin's great-grandson, and a series of evolutionary biologists going around Down House. There I'm with Benjamin Zephaniah Rast, the poet, who came and read some poetry out for us. We made a dub album of The Origin of Species with the Jamaican student. And I also have a cameo role in a rap video. So it was all good fun, and I don't regret it. It was a bit obsessional. I think my wife probably got a bit fed up with it after a while. There we go. Particularly the writing of the rough guide to evolution, which was the thing that really did drive me nutty. 2008, as we're coming towards the end of that decade, there's a particular landmark here from Mark Huffman, who's in the audience, and my old supervisor, Gordon Dugan, where they showed that they kind of went one step further than Zuckerkadel and Pauling, where they're just talking about bits of sequence. They said you can actually use the whole genome of an organism, the whole genome of a bacterium, as a document of its evolutionary history, and you can start to reconstruct the evolutionary history of bacterial species using this kind of approach. And around the same time, this new type of sequencing called high-throughput sequencing came in. So basically, the cost of sequencing plummeted and the ease of use increased dramatically. And I actually, in more jocular moments, said, well, we're going to approach this sequencing singularity, where sequencing becomes so cheap that we can we'll use it for everything. And in fact, shortly after I said that, there was a paper in Nature where some guys at the Sanger had actually taken the complete works of Shakespeare and translated them to DNA sequences and shown that they could store them and then resuscitate them and so forth. The opportunities were opened up greatly also by the availability of these called desktop sequencing instruments about the size of a laser printer. That basically meant we didn't have to work in a large dedicated sequencing center anymore. Any small, medium-sized research group could kind of do this kind of stuff. One area we got into fairly early on was looking at bacterial evolution within hospital outbreaks, looking at an organism called Acinetobacter baumannii in a local hospital, and also with colleagues in Nottingham, an outbreak of Pseudomonas aeruginosa that caused the problem. And we sequenced the whole genome, and we were able to reconstruct some of the transmission events that went on. More recently, we've been looking at a very large outbreak of this organism, Acinetobacter baumannii, which went on for 80 weeks, involved military personnel and civilians to the hospital in Birmingham. And we genome sequenced nearly 100 isolates from that outbreak. And we were able to reconstruct the phylogenetic trees, the evolutionary trees, and the transmission chains, working out who'd given it to whom, where they'd got it from. And then that allowed us to focus our infection control efforts. So it became clear that we couldn't account for everything by patients being in adjacent beds. And towards the latter part of the outbreak, we realized there was an operating theatre at the heart of this, and that cleaning up that operating theatre led to control of the outbreak. Also been involved recently in kind of what you might call Darwin in the drugstore, so Darwinian evolution as a source of antibiotic resistance, where we sequenced 2 isolates from the same patient, same organism, one before treatment, one after treatment, and one before treatment was susceptible to the antibiotic, one after treatment had become resistant. And we sequenced the whole genome and managed to detect some changes between them. And we found surprising, we found a mutation in the gene that was already known to be associated with resistance, but we also found parts of the genome falling out and losing DNA repair. And that was kind of counterintuitive. You kind of think, well, the later things in the outbreak, or in a particular transmission chain, they're going to have more DNA rather than less, but here it was a loss of DNA. And in fact, this is an interesting, many of these themes that we're seeing in terms of sequence evolution, genome evolution, there are parallels here between microbiology, what happens in microbiology, and what actually happens in the evolution of cancers in patients as well, because when a cancer evolves, it starts to lose DNA repair and become much more susceptible to mutagenesis. And we're actually currently following up on this work now. We're starting to look at multi-drug resistant TB isolates from West Africa with colleagues in the Gambia PhD student, my Medicaid single, and with Martin Antonio, one of my former PhD students who now works in the MRC unit in Gambia. 2011, we got involved in what you might call social evolution, where we were basically catalysing evolution with our own discipline. So we, particularly Nick Lohman, who was working with me at the time, we pushed the boundaries where this outbreak had been going on in Germany. The Chinese Genomics Institute, BGI, a genome sequence and isolate from the outbreak, but hadn't really done much analysis on it. It just released the raw data. And then Nick did an initial analysis on this and said, well, these Chinese guys have just released this data without any usage rights or anything. He did his analysis, released it again, and then he called on the rest of the scientific community to help analyse that. And actually what happened within a couple of weeks, there were dozens and dozens of analyses done as a kind of crowdsourced analysis. People on different continents, North America, Africa, Asia, Europe, people in Hong Kong, all using Twitter and blogging and so forth. And so we wrote this up for the New England Journal of Medicine as an example of how things were evolving in terms of methodology. What happened then was we actually were also in collaboration with the German guys who were at the centre of the outbreak. We'd not actually met them when we wrote up that paper, but we went over and saw them. And in fact, I'll put it here, we actually were drinking champagne, celebrating the fact we got this New England Journal paper, and we said, well, what should we do next? How can we top this? Well, let's think about what we have as unique assets. And I said, well, we know how to do genome analysis and maybe metagenome analysis. And they said, well, you've got a freezer full of shit. You've got 200 or more samples from that outbreak. That's a unique resource. I said, yeah, why don't you take your unique resource and start sequencing it? And that was the birth of the next project, where we looked into whether we could just extract the DNA from those fecal samples and sequence it without trying to grow the bacterium first. So in a sense, we were challenging that paradigm that goes back to the 1880s, that basically you have to grow the bacteria as single colonies with microscopy and identify them carefully and then work on them. We said, why don't we just take that sample and sequence the DNA? So we did that, and to cut the long story short, it worked. We managed to reconstruct, Nick particularly with some mental gymnastics, remarkable mental gymnastics, assisted by Chris Quince, who's coming to work here in Warwick in a few months' time. The two of them together basically reconstructed the E. coli genome just from the DNA sequences from the stool samples from the metagenome. And we wrote that up in the Journal of the American Medical Association. And I've written a little review on this called Diagnostic Metagenomics as to how this might go forward. In fact, on the telly a few weeks ago on Midlands Today, saying the same kind of thing. We're also taking this forward now. There's Emma Doughty with Medicate in the Gambia. She's out there currently collecting sputum samples, and we're hoping to use this approach to see if we can get TB genomes directly out of sputum samples without having to grow the organism first. Okay, nearly finished now. So as we're in the last year or so, we've also taken on a new line of work, which is actually seeming to explode, really. We're now looking at ancient and historical DNA samples. And here we're looking at genomes not as documents of evolutionary history, but as documents of human history. So this first came into our lab with this work on TB in a mummy, where we looked at some mummified material, 200-year-old mummified lung, extracted the DNA, sequenced it, and were able to reconstruct the genome of the causative organism Mycobacterium tuberculosis. That work done by Jackie Chan and Martin Sargent with help from Helen Donoghue there. We've also been working with Vince Gaffney, who's sitting at the back there, and Rob Allaby, looking at sediments, 8,000-year-old seafloor sediments, and extracting DNA and sequencing them Rob has actually been leading on the analysis on that work, but it's been interesting to see that work catalysed in ancient DNA. More recently, we've actually found a Brucella melitensis genome from medieval human remains from the early 14th century, and that's actually been that's what you might call vaporware. That paper that I presented there, because it's actually what it will look like if it gets published in PNAS. But we're struggling with PNAS at the moment as to whether they would wish to accept it or not. So it will get published somewhere anyway. And we're now working with Raffaella Bianucchi on other samples. So Gemma Kay, my group, at this moment is over in Hungary collecting material from Egyptian mummies. We hope to get material from mummies from a place called Fiume, where remarkably these mummies actually come in a casket that has a portrait of the individual. So you have these wonderful portraits of these people from the 1st century. And so we could actually get into their metagenomes and recover pathogens. That would be really interesting. I did also point out, though, to Raffaella that the inhabitants of the Mediterranean region haven't changed much over the last few years. They're quite similar. And we're also, she's recently just said we want to look at the remains lung from the first French queen, Aragon, from I think the 6th century. So we've got lots of interesting things going on here in ancient DNA. Another area we started working on since we moved to Warwick, really, is if we try and draw evolutionary trees using sequences and then you apply the Gram stand, sometimes they don't agree. Most of the time they do agree. So gram-positive organisms all belong in one group for the most part. But there is a group within that group of gram-positive organisms that suddenly is now gram-negative, and we can't understand that. It's a real puzzle, and Richard Brown in my group is trying to actually tease that apart and work out how can you explain this in evolutionary terms that a group of gram-positive suddenly become gram-negative. Just the last few slides. So this year, as you heard, we won this MRC funding. I think the key sign that you've arrived is that you're actually important enough to have the piss taken out of you, to be parodied. If any of you are actually still awake and reading that, that's not actually what we won the money for. We actually won $13.9 million to spend on Apple Macintosh computers. And my name isn't Professor Stallion. My name isn't called Dr. Sloman. And he's certainly not the most sexy scientist in Britain. But I am looking forward to we're just kicking this off now, getting started on this project, working with Sam Shepherd there, Tom Connor, Nick Loman, Chris Quince, and there's Mark Ackland. Maybe he's the sexiest microbiologist in the biomedics community, the way he's looking very dapper there at this time. Talking of Mark Ackland, he and I, people thought I was a little bit odd recruiting a 69-year-old to come and work with me, but he's 70 now. At the time I recruited him at 69. But it's borne fruit because we've just won this 1.4 million, 1.6 million grant on Enterobase. But there was a very fraught period, as it always is with these, when you're coming up to the moment when the panel's going to make a decision. So the panel makes its decision, and then that has to get ratified. And in that period between, we knew that the decision had been made, but just needed to be ratified. I actually contacted a panel member, and I was trying to get any clue, and I just turned to Darwin and said, well, this is basically how it's working, isn't it? This is basically, there's a struggle for existence that goes on among grant proposals, just as there is among individuals. And I actually, so geeky am I, actually translated Darwin into the modern grant here. But that actually hopefully brings me back to Darwin, and I'm now just about to finish. Again, you don't want to disappoint the audience, and I guess there's probably a large number of you know how I'm going to finish. I always finish the same way when I give these talks, and that is I'm just going to, because Darwin said it the best, and so I'm just going to close with the closing words of The Origin of Species, where Darwin actually summarised this whole discipline, where there is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one. And that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Thank you very much.